Welcome to this explainer. If you've ever stared at a massive spreadsheet just absolutely packed with tens of thousands of bug counts and thought, how on earth do I turn this into a story about ecosystem health? Well, you are in exactly the right place. Today, we're taking the mystery out of the math. We are going to take 12 kinda daunting ecological diversity indices and turn them into a totally practical, ready-to-use toolkit for your entomofauna field data. We'll break these equations down so they're intuitive, accessible, and honestly, just a whole lot more manageable.
Here is the roadmap for how we're going to do it. First, the rules of field sampling. Then, we look at within-site alpha diversity, followed by between-site beta diversity, and we'll wrap up with interpreting ecological health.
All right, part one, the absolute rules of field sampling. We have to start right at the local scale. So right out of the gate, there's a universal field minimum you absolutely need to hit, 50 to 100 individuals per sampling unit. Why that specific range? Well, you have to satisfy the most mathematically demanding indices, specifically the Shannon-Wiener index and Fisher's alpha. If you can just cross that 50 to 100 threshold, every single other index we're talking about today is going to automatically have enough data to be statistically bulletproof.
To actually capture a well-designed study, you've got to follow these 4 steps. Set 3 to 5 replicate plots per habitat, sample those 50 to 100 individuals, and detect at least 5 distinct species. But step 4 is literally where the magic happens. You have to plot your species rarefaction curve. You keep collecting until that species accumulation curve finally starts to plateau, which proves your sampling is actually adequate. If your curve is still shooting straight up at 100 bugs, you just need more samples, because honestly, no formula is going to save a poorly sampled community.
Moving right along to part 2. Let's crunch some numbers on within-site alpha diversity.
Alpha diversity is entirely about what's going on inside a single isolated site. It's asking what is happening right here, in this exact patch of forest or this specific stream. We're mathematically examining the richness, the dominance of certain species, and the overall evenness of your specific bug community.
But before we dive into the formulas themselves, we absolutely need to get how these numbers actually behave. We basically split these indices into 2 camps. Bounded indices scale universally from 0 to 1, providing these really nice, universally agreed upon hard thresholds for ecological disturbance. Unbounded indices, on the other hand, stretch anywhere from 0 to infinity. They don't have universal cutoffs because their values depend entirely on the specific organism group and geographic context you're working in.
Let's look at the 3 unbounded richness indices. Simple species richness, or S, is literally just counting the species. It's super simple, right? But it's heavily biased by how much sampling you actually did. And that is why Margalef's and Menonick's formulas are so clever. They elegantly fix that sample size bias by dividing your species count by either the natural log or the square root of the total individuals you collected. It is a brilliant little mathematical trick to level the playing field if you're trying to compare a massive bug catch to a much smaller one.
Okay, so what do bounded indices actually tell us about health? Well, the Shannon-Wiener index measures the uncertainty of predicting a random bug's identity, usually sitting between 1.5 and 3.5 in natural communities. Then there's Simpson's dominance index. It scales from 0 to 1, and a higher number means a single species is completely taking over, which is a massive red flag. Its direct opposite is Simpson's diversity index, where a number closer to 1 means you have great diversity. And finally, PL's evenness tells us how equitably individuals are spread out across the species present. If that drops below 0.4, it suggests severe ecological disturbance. These single community indices are absolutely at their best when you use them all together, kind of like a dashboard.
Rounding out the alpha indices, we have 2 really fascinating outliers. The Berger-Parker index is just a delightfully simple measure of single species dominance. You just take the abundance of your most dominant bug and divide it by the total catch. Super easy. Meanwhile, Fisher's alpha is a completely different beast. It's a complex log series distribution parameter, but its massive superpower is that it is incredibly stable across wildly different sample sizes. It's absolutely perfect for comparing field efforts that were a little uneven.
All right, on to part 3. Let's zoom out to between-site beta diversity and do some comparative math. Beta diversity is all about comparison. It lets us mathematically evaluate how our insect communities are shifting when we walk from one habitat to the next. So, are the beetles in plot A totally different from the beetles in plot B? Or are we looking at the exact same community, just living a little further down the road?
When we want to compare overlap, we turn to similarity indices, the big ones being Sorensen's and Jacquard's. Both measure similarity on a nice, bounded scale from 0 to 1, but ecologists generally prefer Sorensen. Why?
because the formula actually gives double weight to the species that are present in both sites. That simply provides a much more robust, realistic picture of true ecological similarity.
Hitting our widest scale, we reach gamma diversity. Gamma is the whole shebang, the total regional species richness of every distinct species across all your sites combined. And Whitaker's beta formula elegantly takes that regional gamma baseline, divides it by the mean local richness, and boom, it calculates the true species turnover across your entire landscape. Finally, part 4. Let's translate these numbers by interpreting ecological health to get some real, actionable insights.
Look, all these formulas are pretty much useless without context, so let's ground them. Whether you're standing in a tropical rainforest canopy or wading through a temperate stream bed, ecologists generally agree on some universal red flags. If your Pelo's evenness drops below 0.4, or your Simpson's dominance climbs above 0.5, or that Simpson diversity 1-D drops below 0.3, mathematically you are looking at a highly disturbed, degraded, or aggressively dominated community. These thresholds are a giant warning sign.
But when you pivot back to unbounded indices like Margalef or Fisher's alpha, you have to remember this absolute golden rule. The organism group matters enormously. The exact same patch of woods is going to give you wildly different numbers for trees compared to ground beetles. You know, a published baseline for bird communities means absolutely nothing for macroinvertebrates. You always have to compare your unbounded numbers against pristine, undisturbed reference sites for your specific taxonomic group.
I really hope this explainer has successfully transformed these complex formulas from intimidating math into a super practical toolkit for your entomofauna research. You've got the field rules for valid data, the alpha measures for local sites, and the beta metrics to compare regions. Your toolkit is fully assembled. But the big question is, which of these 12 indices is going to be the key to unlocking the true health of your next field site? Seriously, when you run that next rarefaction curve and crunch these numbers, what kind of hidden story is your data going to reveal about the ecosystem's true health? Thank you so much for joining me on this deep dive. For more clear, structured breakdowns of ecological science, make sure to like and subscribe to the Entomofauna channel. Keep exploring, keep sampling, and I'll see you in the next explainer.